Well, hello everybody. My name is Lee Rowen, and I was invited by Stan to conduct this conversation with my friend and mentor, longstanding, Lee Hood, so it's my pleasure to do that. So let's begin. Lee, you grew up in rural Montana, and then from there you did undergrad at Caltech, and then from there you went to grad school, and then to medical school.
I went to medical school first.
You went to medical school first,
Yes.
at Johns Hopkins, and then to grad school, and then took a faculty appointment at Caltech. So what or who were the significant influences on that high trajectory career path?
Well, I think among the most significant influences were those that occurred actually in high school in Montana. So I went to a small high school that had about 140 students in it. But it had three of the best teachers I ever had. One in mathematics, one in chemistry, and one in social sciences and history. And they gave me an entirely different perspective at what it meant to be educated. And in fact, the chemistry teacher had gone to Caltech during World War II in the Navy as a navigator. And so he decided any good students he got, he would really direct toward Caltech. And I have to say, I'd never heard of Caltech, but he insisted I consider it. I did, and it was a wonderful place for me to go. The second thing that happened to me in high school that was really interesting is my father was an electrical engineer, and he ran courses in circuit design, circuit theory, systems theory, and things like that for people who worked for him. And he liked to have me take the courses with his men. Now, I really, at the time, hated these courses, but I will say they really influenced how I thought about biology in later years. And the final experience I had in high school was my grandfather ran a geology camp in southwestern Montana in the Bitterroot Mountains, Beartooth Mountains, for students from Columbia, Princeton, Harvard, Yale, and I got to take courses in geology with some of those students. In fact, did a project that took me back to the Westinghouse Science Talent Search in Washington, D.C. So I got a really spectacular start in high school. And I would say Caltech had absolutely wonderful faculty, and they were all enormously accessible, and you could do science. But what I think was really exceptional about Caltech were the students. I mean, they were incredible students. I mean, all of them enormously talented. We had a mathematician in my class that could work chemistry and biology problems better than any of the chemists or biologists could. And it was those really exceptional people that set a very, very high standard. And at Caltech, I began to realize what I was really interested in was medical science. And Caltech was Renato Del Becco and Max Delbruck, so it was all microbial biology, molecular biology as it existed in those days. So that was the reason I decided to go to medical school at Hopkins. And Hopkins, fortunately, had this accelerated program where you could finish in a little less than 3 years if you went right straight through. So you took all the clerkships and everything else, but I finished that up and then went back to Caltech as a graduate student for 4 years, before then going actually to the National Institutes of Health. I'd really wanted to go abroad and do a postdoc, but it was the Vietnam days, and it was clear that anybody with an MD degree would go to Vietnam if they didn't go to the Public Health Service or the Indian Service or something like that. And that was a wonderful preparation for my career, because in those years, I think almost anybody who was anyone in American medicine went through there. So in 3 years, I got to meet the people who were the kind of the future leaders in American medicine. And then, of course, I looked at where to go back to school after that and get a PhD. And I'd say the two places I really considered seriously were Caltech and Stanford. In fact, I remember interviewing for a job in the chair of microbiology's office at University of Colorado. And Bob Sinsheimer, the chair of biology, called me up there and offered me a job over the phone in this guy's office. And the guy actually predicted the whole thing, and he said, well, we can have a good time now because I know you're going to go to Caltech. We're not going to worry about this anymore. And I did. I went to Caltech.
So when you took your Caltech appointment, you said that you wanted to devote half of your time to biology and half of your time to technology. And I imagine this was influenced by your graduate advisor. But why was that? Because that would have been kind of revolutionary at that time. So what were you thinking when you said this half-half thing?
Well, another person that did really influence me was my graduate advisor, Bill Dreyer. And Bill was dyslexic and incredibly creative in many different ways. And he left me with two fundamental tenets. One was always practice biology at the leading edge because it's more exciting there. And the other was, if you really want to change biology, invent a new technology and explore new dimensions of data space. And I took those dictates very seriously. And in fact, I got offered a very nice position at NIH, but it was utterly clear it would have been very difficult in those days to develop technologies at NIH, and that was one of the reasons I ended up choosing Caltech. And philosophically, so I started working in molecular immunology as a graduate student, and what I gradually became aware of, and Max Delbruck was the one who really drove this home after I'd been at Caltech for a while, was that the immune system was really, really complicated. And I began to wonder whether one gene and one protein at a time was really going to take us very far in immunology. And I will say what we did in molecular immunology, that is deciphering how antibodies diversified themselves, was in fact a biological phenomena that was really susceptible to very simple approaches and so forth. And over a period of 10 years, we really delineated a lot of what that was all about. But the real phenomena in immunology were the immune response, tolerance, autoimmunity, and it was clear we didn't really have the faintest clue what those were all about. And in fact, in many ways, we still have lots and lots of questions as to what those are about. And it was those phenomena. I mean, Max Delbruck made that argument to me in the late 70s. He, by that time, decided to take up the study of phycomyces, and he decided he was going to do it from a systems point of view. And he talked a lot about that. Now, there weren't very good tools for doing things from a systems point of view then. But he pointed out that immunology was going to be impossible unless you started thinking about it more of a systems way. And but anyway, what drove me to Caltech and doing technology and biology was just this idea that the two could interplay off one another very nicely, that the biology, if you did frontier biology, you needed new tools to be able to figure out what it was all about. And once you got the new tools, that really revolutionized the biology. So that kind of started me on this dual course that on the one hand led to the development ultimately of 5 instruments for synthesizing and sequencing proteins and DNA. And on the other hand, it got me involved in molecular immunology and neurobiology, and then started pointing my interests more and more toward medicine, which I was able to really start exploring when I moved to the University of Washington sometime later.
How do you make the judgment call as to when a problem is tractable, especially if you're developing new technologies to attempt to solve it, versus, look, you can develop these new technologies and they might be good for solving something else, but dude, you picked the wrong problem because it's not tractable? How do you make that judgment call?
You know, I've been asked that question before, and my answer is I don't think it was ever a conscious choice. I think solving one set of problems led to another set of opportunities, and it's certainly a judgment call about which opportunities you explore and so forth. But I'm a determined optimist, so I always thought I'd be able to figure out how to do things, and so I don't think I really worried about to what extent problems were really tractable, except in the sense of biological complexity and how we were going to deal with it. And there, fortunately, the first course I took was one of the most fundamental ways we have to deal with complexity. That is, biological complexity in part comes about as a consequence of the random and chaotic process of Darwinian evolution. And, complexity builds on complexity, and you end up with systems that are Rube Goldberg-like in both the complexity of their elements and their associations with one another. So the question is, how can you figure out what the elements are? How can you figure out what their connectivities are? And how can you figure out the dynamics of how they operated? And those were things that I started thinking about probably in the 80s, but it was clear we couldn't do very much about it. We didn't have good technologies, but we didn't really have something that became later really important, a complete parts list of all the human genes. We only had a partial list, and you always operate at a disadvantage. I mean, I think the analogy of how an engineer would figure out how a radio works is really a good analogy to how we have to work on biology from a systems point of view. So the first thing the engineer would do would be to take the radio apart and study all the components individually and see what they did. And, that was biology for 40 years or so in the molecular biology era. And what is utterly clear is the studying of those individual components would never give you the faintest clue of how the radio worked. What you had to do was put the components together in their circuits and learn how the circuits operated individually and collectively to be able to convert electromagnetic waves into sound waves and so forth. And I think it's exactly the same with biology. We needed to have the tools for generating a lot of information, but the next thing we needed really was this parts list so we could begin to think about how to do holistic global analyses of systems and so forth. And of course, that all came somewhat later when there was a convergence of a lot of different things that were important. But of course, one of the so I'd say, in a sense, I've been involved in a number of interesting paradigm changes, and the first was, in some ways, bringing engineering to biology. And I think the important paradigm change there was just the idea that you could generate technologies that could create enormous amounts of information. And we see it in spades today in next generation DNA sequencing. And then the next paradigm change that came, and in fact it was my participation in the second one was triggered in fact by the developing the automated DNA sequencer in the early and mid 1980s. Bob Sinchheimer had the first official meeting, anyway, of the Human Genome Project at Santa Cruz in the spring of 1985. And he was very interested in exploring the possibility of setting up an institute to sequence the human genome. So he invited 12 specialists in this area. I was one because of the technology aspect, but Wally Gilbert was there, George Church was there, David Bodstein was there, and Ellen Donis Keller. I mean, and it was a really interesting 2-day debate, and we ended up coming to, I thought, 2 conclusions. So one was, it was feasible, although really difficult, 85. If we used the methods we were using then, it probably would have taken 100 years to sequence the genome. But the second thing that was evident was there was a split 6-6 in the people that were there on whether it was a good idea or a bad idea. And the people who thought it was a bad idea were really vehement about it. And they thought it was a bad idea. Big science is bad science. The genome is mostly junk. Why would you want to sequence it? Sequencing the genome is work for monkeys. How could you ever get anybody very good to do it? And those were the kind of arguments that we ran into. And then, in fact, from 85 to 88 or 89 in the community, certainly the bulk of the biologists were really bitterly opposed to this project. And in fact, the National Institutes of Health was bitterly opposed to this project. DOE was actually the agency that really pushed these kinds of ideas early on. And the pivotal point there, of course, was a National Academy committee that was set up, headed by Bruce Alberts. And it was more or less half opponents and half proponents of the Genome Project. And it unanimously reported back that this should be done. And that kind of turned the tide, and the project got started in 1990. And it's, if you think about it, it's really a remarkable project. And I think it's been one of the most transformational projects in all of biology. I remember Nicholas Wade wrote a New York Times article, about a year and a half ago, on the disappointment with the Genome Project and what it had really generated. And I sat down and wrote about 15 things the Genome Project did that were good. And, they were, number one, the parts list that made systems biology possible. Number two, the Genome Project really democratized genes. By that, I mean it made all genes accessible to all biologists in a way that never could have been done otherwise. The Genome Project really pushed technology development, and not just of the genome kinds of things, but much broader into proteomics and other things as well. And I think the Genome Project, in many ways, was the project that was really responsible for providing a secure home for computer scientists and software engineers and all these other people that were desperately needed to do this. And it was one of the first projects that worried about the validation and quality of data. It was one of the first projects that utterly insisted that the very smallest fragments of DNA should be available to all scientists, that is, open data and so forth. And of course, it made the genomes of microbes and plants and animals accessible, and in doing so, revolutionized a bunch of fields. I think it utterly transformed the field of evolution in just striking ways. And it did a whole series of things for medicine. It certainly was a wonderful first example of big science in biology. And it later led us to appreciate that there are many different types of big science, and some are good and some aren't so good. But it was one of the first projects that really spent a fraction of its thumb on ethical, legal, social kinds of things. And there was a a recent estimate by the Battelle Institute, and I don't know if you...
I've got it. Okay, ready?
want to read it?
Sure. The 3.8 billion put into mapping the human genome between 1988 and 2003 helped support approximately 310,000 jobs, 100,000 jobs here, that produced 20 billion in personal income in 2010 alone, according to this Battelle. The report lauded investments in the Human Genome Project as some of the United States government's greatest in terms of their return. And then if you think about it, the whole up-and-coming industry that's going to arise in the area of personalized genomics, consumer genomics, personalized medicine, who is going to be staggering in terms of the impact on healthcare and employment and money and so on and so forth. But let's go back in history a bit beyond, that's the future. Okay, so here you are bebopping along quite nicely at Caltech, developing novel instrumentation, yet you left and you came to University of Washington to form the molecular biotechnology department. And so there's a quote that you've made that says, new visions need new organizational structures to actualize them. So my question is, what were your hopes for the molecular biotechnology department, and what did you hope to actualize that couldn't be actualized at Caltech?
Okay, well, let me give you a little context for answering that question. One of the things that came out of developing the automated DNA sequencer was, well, the fact was we spent 3 years trying to automate it with a really good biologist and totally failed. And I put together a group that had a good engineer, a really good chemist, a computer scientist, and a molecular biologist. And in about 6 weeks, we'd figured out the basic logic, and then 3 years later, we developed the prototype instrument. But what that really said to me is that a lot of the frontier problems in biology are going to demand, in the future, developing very sophisticated technologies. And hence, you need to create environments that are cross-disciplinary in nature. So again, this idea of biology driving technology, and in addition to it, technology driving analytic tools could actually be fulfilled and realized. So I went to Tom Everhart, who was the president of Caltech in about 1988 or 89, and I proposed to him that I set up a new Department of Biology, molecular biotechnology at Caltech that was to have this cross-disciplinary character. And he said, well, he'd be in favor of it if you could get the approval of your colleagues. So I got the approval of the engineers and the chemists. I think the physicists were pretty indifferent, but the biologists were really opposed to it at Caltech. And one of the characteristics about Caltech, which has absolutely superb biology, is they really were against, quote, this big kind of biology. And when they turned down that request to try something new, I realized that if I wanted to do these kinds of things, I wasn't ever going to be able to do them at Caltech. And I did go out and look at several places. I looked really very carefully at Berkeley. And then through a series of chance circumstances, was up visiting Roger Perlmutter at the University of Washington, and I talked a little bit about the kind of things I was thinking about. And he really catalyzed a lot of things by bringing this in front of the dean, and then getting Bill Gates at least interested in this kind of thing. And I got invited at that time to give a series of lectures up here. Is it the Dreyer lectures or no?
I don't remember the name of that.
But anyway, Bill came to all three of the lectures I gave.
Walker Ames lectures, I think.
Yeah. No, it wasn't the Walker Ames.
John Dance.
That might have been those. Anyway, whatever they were, Bill came to all three of them, and then we had dinner atop the Columbia Tower for 3 hours or so after the third lecture. And he said, well, what would it take to have you come? And at first he said, really, what would you like to do? Where would you like to go? And so it was Bill that really made it possible to consider coming. He gave a $12 million gift as the centerpiece of getting this new Department of Molecular Biotechnology started. And that made it possible to move a number of the people that were associated with me at Caltech, some of whom are still here. And then we started recruiting other people, Maynard Olson and Phil Green, and some of my former students, John Yates, Rudy Abersol. I mean, it was a terrific list of people. And the department over a period of 8 years was incredibly successful. And the hope that I had was that one would build this cross-disciplinary infrastructure and then on top of it be able to create a department of, or a group of systems biology. And Phil Fialco really liked that idea, but as you all know, Phil died about halfway into that plan on this avalanche in the Himalayas. And I found after that point in time that it was challenging to do the things I felt needed to be done for systems biology. For example, I remember talking with the computer science people here that ran computer science at the University of Washington, and really arguing with them. They felt a small janitor's closet would be more than enough for all the computational needs of any department of biology, and they were undissuadable about the fact that the needs were going to become infinitely larger in the future. And there was the need to build up these emerging kinds of technologies, and again, getting the resources to do that, and frankly, the space to do that in some cases was a challenge. And it was those kinds of things that we struggled with for a period of 4 years before I finally decided, gee, if we were ever going to start this department, we'd have to start afresh. And I, in 2000 and late 1999, resigned from the University of Washington to start the Department of Systems Biology. But I will say, in looking back about the Department of Molecular Biotechnology, I mean, in some ways it was really remarkable. So Phil wrote some of the key software for the Genome Project, assembly and quality control. Ger van den Ing invented a multi-parameter high-speed cell sorter that kind of set the pace for the last 10 or 15 years in these machines. We actually here developed the fifth of the machines I developed, which was the inkjet DNA printer, which is what Agilent has now employed to synthesize DNA, to make arrays, to do those kinds of things. And of course, Rudy Abersol and John Yates were among the very first pioneers in proteomics. They developed one computational and one chemical technique that were the early foundation of proteomics. And of course, the department, in a sense, had two genome centers. Maynard had led one center, and Lee and I led another center, and I think we made enormous contributions there. So the department was enormously successful, but I really wanted to push it to the next level of bringing in this holistic, integrative systems biology, but to have it built on a framework where the biology could drive the technology and in turn the new analytic tools and so forth. And of course, this department was a part of the precursor of the assembly between genetics and molecular biotechnology that led to genome sciences. And I suspect in many ways the most important contribution we made are the wonderful people that came out of this department to go into to genome sciences.
If you can reflect, since it might be of use to this department, what do you see as the most essential enabling conditions to support cross-disciplinary science of the sort you think biology needs to have?
Well, I would say, one, there has to be a will to bring in very different types of scientists, and then a commitment to essentially having them learn one another's language, and a commitment to having them learn to work effectively together in teams. I think for us, that has been one of the enormous challenges. I think number two, it is really important to have the high throughput technologies you need that are kind of the fuel for systems biology. And in fact, I think one of the things we've achieved in that regard is what I would call the democratization of data generation and data analysis tools. And by that I mean at ISB, a scientist can use those things to do big and or small science. It isn't that you have to learn how to run a lot of this sophisticated machinery, but you have access to them in a way that you can drive your own research projects. And I think finally what is critically important in creating anyway a stable environment is to realize that in the end, systems biology is biology. It has to be driven by the biology. The biology, as I said, mandates the technologies you develop, the analytic tools you need. But as I look around the world at the 60 or 70 centers for systems biology or institutes for systems biology, I see a lot of interesting flavors. I see a subset of them where there are primarily theoretical computational people. And I think those are in a hopeless position because biology is so complicated. Unless you have close contact with the real data, you're not going to approximate the reality of Darwinian evolution for systems. I see another set that are utterly enamored with generating enormous amounts of data. And again, that alone isn't sufficient. I mean, data, large data sets, I think pose two enormous problems. So one is the problem of signal to noise, and the other is the even bigger problem of how you convert data into knowledge. And I would say that systems biology really gives us the fundamental strategies and tools for attacking effectively both of those major problems. And I think that's what a lot of people who generate a lot of data don't really understand. And an essential part of the strategy for dealing with signal to noise is, one, you really have to capture the dynamics of systems. If you capture the dynamics, you really can get rid of a lot of the peripheral noise and things. Number two, I think it's really important to be able to effectively integrate different levels of data together in such a manner that you can actually make predictive models and for medicine, actionable models, ones that will give you hypotheses who will improve your patient's health and so forth. So, and so I think what is ideal is to have these be biology driven, but to have the people that can approach these fundamental problems from the computational mathematical side as well as from the experimental side, and each keep in close contact with one another to keep you on the right track and everything.
So how important do you think strategic partnerships with industry or collaborative other institutions or whatever are to the success of this complicated field of biology, especially in this era of difficult resource allocation?
So, so I think, I think it's interesting. One of the grand challenges in the 21st century, I think for every discipline, both engineering and scientific, is complexity. And I think biology is unique, perhaps because it has really effective systems-driven approaches for dealing with its complexity. I mean, thinking about biology as an informational science, that sets up the logic for how you can deal with noise really effectively. Setting up this infrastructure that gives you access to tools that can generate and analyze data and develop new kinds of technologies. Using experimental systems approaches that can capture dynamics is really important. Capturing dynamics is one of the critical reasons why model organisms are so important. You can't do dynamics in humans except in very, very limited ways. So the challenge then, if you're going to use a model organism, is how do you match that model organism's disease against a human disease, or its biology against a human's biology? And that is, as we all know, a real challenge. And of course, I think one thing that's really important for deciphering complexity is to really encourage emerging technologies. So we all know about genome sequencing and how that's exploding, but it's exploding in many ways. It's letting us do biology in new and interesting and more powerful ways. I think the approaches that have been taken where you integrate together genetics and genomics through things like family genome sequencing offer real advantages in dealing with signal-to-noise problems. I mean, in proteomics, I think the field of proteomics is really going to be revolutionized in the next 5 years. And what ISB has been responsible for doing in the past 5 years, together with Rudy Abersol, is creating this field of targeted proteomics. And the bottom line there is we now have assays that can be assessed by mass spectrometry for virtually all human proteins, and a database that basically has those assays. But the one thing that proteomics is lacking that genomics has is the ability to do global analyses, because in the end, they too are really key to systems biology. And there are new approaches that, together with Rudy, we're beginning to develop, where we really think we can make proteomics global within the limits of sensitivity of the mass spectrometer. And the really attractive idea is there is a new form of mass spectrometer which can capture the digital information from all of the tryptic peptides in any particular proteomics mixture you'd like to do in an hour. And once you've digitalized that, you have that data for the rest of your life. And the idea then is to set up a standard database that has most of the major tryptic peptides profiled in mass spec space. So then you can take any peptide here and find its analog in the standard, and from the standard you know what it is, so you can search. And as we fill out the standard, we'll just be able to do more and more global analyses. And what's really exciting to me is then we can do dynamics not only at the level of genomics, transcripts and microRNAs and things, but we'll be able to do an impedance match at the level of proteomics as well. Single cell analysis, I think, is absolutely going to transform biology in a lot of ways. I think there are incredible ways you can use iPS cells both to discover fundamental new aspects of biology, development, physiologic responses, and to actually do very powerful diagnostics, ways you can use them to stratify disease and things like that. So all of those emerging technologies are just transforming what we can think about doing in systems biology. And then, of course, there are a parallel set of analytic tools that you have to do to be able to handle the information and process it and so forth. And for me, what's been exciting is to take these very powerful pillars that we can use to deal with complexity and then point them at big problems in science. So, I mean, what's interesting is that if you think about the big problems that society has today, healthcare and global health and energy and the environment and nutrition and animal health and all, I would say each of those problems can be attacked in exactly the same way with this kind of systems thinking to what we've done for biology on the one hand and to a certain extent healthcare on the other hand. And of course, one of the interesting questions for any academic organization is, what do you have to do to create an environment that will enable you to do these things really effectively? And that, I think, gets back to the cross-disciplinary environment and this holy trinity of biology driving technology, driving computation, and democratization of data generation and data production tools and so forth.
So one of the things that can contribute to the cross-disciplinary is partnerships between academia and industry, because the commercialization enterprise can actually make a product so much better than what the academics produce.
Yeah, so I agree. I mean, I about 5 or 6 years ago started thinking about strategic partnerships, and my thoughts were, what is attractive about a strategic partnership is you can use it to attack these big problems. So one, you can select out partners that have complementary skills and technologies, expertise, and so forth. Number two, some strategic partnerships open up completely new approaches to funding that are very, very big compared to the smaller grants you usually can get from federal grants and contracts and so forth. A third idea that really is attractive to me is with a strategic partnership, it can be internationalized. That is, you can say, who are the best people we could get to solve this problem? And you can go and propose a strategic partnership to those. So ISB I've spent a lot of time in the last 6 or 7 years on strategic partnerships, and we've developed strategic partnerships, at the level of a country in one case, Luxembourg, certainly at the level of academic centers and clinical centers on the other hand, and at the level of companies on the third hand. And I think partnerships are always necessary for really big problems, because one thing it enables you to do is to create a milestone-driven approach that is highly integrative of all the things you need to solve the problem. And I think, again, R01 science really does beautiful things in solving specific, concrete, tangible problems. But with these big problems, you really do need these more integrated approaches if you're going to put them together. And this thing that we've been pushing, this proactive P4 medicine, is certainly a very nice example of that.
Okay, I have one last question, and then we'll open this up to any of you that have questions for Lee. So my last question is, you haven't done it tonight, but oftentimes I've heard you say, well, within 5 years we're going to have solved XYZ, or within 10 years we're going to have biomarkers for this, that, and whatever. So my question for you is, imagine that you're reincarnated 100 years from now, and all of your pressing healthcare problems are solved. What are you going to work on when you grow up, when you're born in 2112?
Well, I think probably the final grand challenge is consciousness, and certainly aspects of the mind that I don't see us being actually very close to approaching. I mean, we can break something and we can see major changes, but whether we really understand those things in terms of the nervous system, I don't know. But it does remind me of a story. You know, I remember the time in 1998, I was asked to go to this big conference and give a projection just 50 years into the future. So what I did was went 50 years backwards, and I got out a bunch of textbooks and saw what the state of the knowledge was. And I realized that the majority of the textbooks thought proteins were the genetic material. And my point was, if you're going to project that far into the future and you have a fundamental error in something like what the genetic material is, you don't have a chance of making predictions that are very good. But you can make good ones for 10 years. The great thing about 10-year predictions is you can, over a period of time, continue to say in 10 years that. So you fudge, you can fudge a little bit on when you actually get there.
Okay, so do any of you have questions for Lee?
Lee, what do you think the ISB is doing with graduate students that might reflect this new view of biology? Are there lessons that you've discovered that could be applicable at the UW?
Well, I think, so I'll tell you my own feeling about graduate students is I think it's really important that they get a cross-disciplinary training. And in fact, at the undergraduate level, I'd argue everybody should have a dual major, biology and something orthogonal. And, you know, as you know, there are a lot of people who would passionately say, well, we just have barely enough time to teach them one thing now. But my experience, even at Caltech with most of the courses I took, is they were courses given by people who were experts in the material and had infinite detail, and you forgot 99% of it within 6 months. So suppose we got rid of a lot of those kind of courses and really concentrated on kind of systems approaches to things and on conceptual approaches to things and on, you know, I think what we haven't really thought enough about is how do we make our students really be analytic? I mean, I think physics and the hard sciences do it through working lots of problems, and maybe we can kind of do it through journal clubs, but that's only kind of. But I think even more fundamental is the question of how do you get people to think out of the box? I mean, as I look across the kind of paradigm changes that we've talked about, you know, bringing engineering to biology and the genome and cross-disciplinarity and systems biology and P4 medicine, what I see in every instance is infinite skepticism and really an unwillingness to kind of openly think about new ideas. So I think a really interesting question is how do we train our students to be open? And I think computer games actually really have some enormous opportunities for doing that thing. I think the kind of training, so we haven't focused in the same way you do on the training of graduate students, but I think what we've done that's very good is we do really encourage them to be very cross-disciplinary, to learn to do things beyond just what their own project is focused on, and to work together in groups where they see people with other talents bringing together their skills and so forth. So, and you know, I think that's going to be the currency for grad students in the future, because I think things, I mean, I guess the other thing that I would say is I think what most biology students don't have, unless they come in from the computational or physical sciences, is good mathematics. And I think not having good mathematics is really going to penalize students of the future.
When you were coming here in, was it 92?
92.
Yeah. if the vote had been taken of the local biologists as to whether you should come, you would have not come. Because I can remember a large number of people, and I was one of them, to be honest, saying, we don't want this guy here. He's the 800-pound gorilla. He's going to come here and lie down wherever he wants, and we're going to get squashed. Did you sense that, and how did you deal with it?
Well, I'll tell you, that's really interesting. When I went to Berkeley, I was going to have a lab with the national labs there and a lab down on the campus themselves. But they did that. They took a vote on whether it was a good idea to bring me or not. And the vote, as I recall, was about 60% in favor and 40% against. And that was certainly a real downer. I mean, that was not the real reason I didn't end up going to Berkeley, but that was a sobering aspect of it. Look, I think if you took a vote at Caltech about whether it was good, whether I was there or not, I think you'd get a lot of skeptics in biology there too, because their view of biology was really quite different of my view for biology. How do you deal with this? I think there are two ways you deal with it. The most important way is to show the vision you have really can change how science is done. And I mean, that's in the end what really counts. I think the other aspect of it is, and we really tried to do that when we were at the UW, was I think big science and small science can make wonderful partnerships. And I think each enable the others in very powerful ways. We tried to do that to a certain extent. But as I said, in all 5 of these paradigm changes, there were always enormous skeptics. So I guess I've kind of gotten used to skeptics and really have tried to think about winning them over by just making sure we could do what we were going to do, basically. Yeah.
Lee, you've talked about some of your high school teachers. Who were some of the influences along the way during your professional career? Really important colleagues who you would discuss things with.
So Bill Dreyer really fell in that category at Caltech. Ray Owen was an immunologist at Caltech who really should have won the Nobel Prize for his marvelous discoveries of tolerance in 1944, 1945. And he was an enormously wise mentor and a real and had an enormous sympathy for a lot of the things I wanted to do, even though I think in some ways he didn't really understand it. I had really interesting interactions with Max Delbruck, and he always made perceptive, insightful comments. I mean, he had an incredible reputation among seminar speakers at Caltech. If he didn't like the seminar speaker, he would walk out of the seminar, write down the projector so his shadow was cast on the, and that was a statement that I remember inviting a really famous immunologist, and he lasted about 5 minutes, and he did that, and I thought, oh God, that's so. At medical school, the person I think who most influenced me was William Wood, who was the youngest ever chair of medicine at the time. He was the chair of medicine at WashU, and then he later came to Hopkins. I don't know, maybe some of you know Bill Wood, who is a developmental biologist now at the University of Colorado. And he had really a very big vision of what science was and how people should think about science. And he was enormously tolerant in that regard too. I would say at NIH, there were really incredible people there to talk to when I went. Gordy Tompkins was there. He hadn't yet gone to UCSF. Chris Anfinson was there, and I got to know these people. And there were a whole series. Actually, my younger colleagues at NIH were really impressive. Gil Oman was there with me at the time, and many others who went out to become leaders in medicine. I would say, without a doubt, when I went back to Caltech as a faculty member, the person by far that had the biggest impact on me was Eric Davidson. And I would say just flatly, of all the scientists I know, Eric Davidson, I think, is the best systems biologist. And I think actually the work he's done in the last 10 years is right at the top of the pack in doing systems biology. But he was always just enormously hard-nosed, very, very data-driven, but enormously open to new ideas. He came in hating technology, and after we'd been together for 5 or 10 years, he ended up really embracing it. So we really learned an enormous amount from one another. Here at the UW, I really appreciated Phil Fialco. I thought he had a lot of wisdom and insight in terms of collaborations. Paul Lang, chief of urology here, really got me into cancer, and that was very exciting. Lane Ostrander, we did some really nice work on genetics of prostate cancer. And some of the colleagues in the department. I mean, Maynard, in his own inimitable way, really made a big impact on my thinking. And it was wonderful to see Rudy Ebersol and John Yates just kind of blossom out and take leadership roles in proteomics. In fact, I would say those two are among the 3 top people in proteomics in the world today. I mean, they're really, really world class. It was really an exceptional group. Gerrit van den Eng, I don't know how many of you know him, but he's an incredible character that sees life in very different ways than most of us do. But it was almost always refreshing, so forth. So,
Lee, it seems to me, listening to you give this really interesting, great talk, that the places you've been, like Caltech, in my mind, they emphasize a department in which you had a series of individuals who do their own thing, and there's a tremendous amount of interaction. And I know Herschel Rowen, who started the Department of Genetics here, that was his ideal in building this department. But you seem to have gone ahead in what you wanted to produce, something which is not quite the opposite of that, but to bring individuals who would interact together, work on a product. And it seems to me that is sort of not quite the ideal sort of academic approach. And there's more something like maybe what you've gone into now with your systems system. Is that the way you feel you've evolved?
well, I think I've evolved along these lines, driven by being impressed with how complex everything is and trying to develop tools and strategies that will let us deal with this complexity, will let us generate lots of data, will let us deal with signal to noise, let us think about ways that we can model this information so you can make predictions. So my own feeling is that there is a place for both types of science, big science and small science. And as I said, I think they can be enormously synergistic and interactive. And people can start out on one side and very naturally evolve into other, depending on what their inclinations are. But I think I will say, in the midst of big science and taking on big problems, you can carve out for individuals, whether they're faculty or grad students, really beautiful problems that are their own. That is, you don't need 40 people to solve a problem that revolves around a new approach to diagnostics and something. If you have either technologies or computational tools that you can develop. So the big science is a compilation of a lot of little sciences that get welded together, and in the case where the problems are hard, you integrate, but many of the faculty at ISB are doing small science too. I mean, they're working on their own individual projects. So I don't know that there's really, I'll tell you, we aren't going to solve the hard problems if we only had small science. I'll guarantee you that. And that's what really worries me about the attitude. Every time the budgets get tight, the small scientists come out of the woodworks and would like to do totally away with big science. And in fact, I was on a committee set up by General Medical Sciences about 4 months ago that considered some of these glue projects, big science projects. And again, it took me right back to the genome days. The hostility against big science was just utterly blatant on that committee. And you couldn't tell them that big science was worthwhile. Although I think by objective criteria, of the 6 projects, 2 did really well, 2 did okay, and 2 did really poorly. And I'll tell you, if you go to R01 grants, I don't doubt you're going to have much different record than that there. And I pointed out to them, gee, has anyone ever looked at R01 science? What about the idea that we have 150 labs doing P53 studies? Is that really the most effective way to do science and spend our monies? But I think the real point is they're synergistic and mutually cooperative, and I think both really important.
Looking back at your career, one thing I don't understand very well, and that is your detour into medical school.
Medical school?
To medical school. Did you expect to go into practice?
No.
Or were you, and why medical school instead of a PhD then?
I went to medical school because I was really convinced I was interested in human biology and human disease. And what I planned to do initially was just take the first 2 years, the courses, and then go to graduate school after that. But Hopkins, as I said, had this accelerated program where you could do everything in a little under 3 years and get finished. So I did that, but immediately after that I went back to Caltech and got my PhD. And I will say, I think that is the right order to do MD PhDs. What I found really nice about it is, one, you could think about your PhD in the context of a deeper medical knowledge, but number 2, you had the continuity all the way through, and you didn't get your PhD, then you finished your MD, then you did your residency if you were going to go on. It's a bit schizophrenic the way it is now.
But it'd be hard to go back and do a residency after your PhD, I bet.
People have done it. You know, I don't think it's that hard. But anyway, that's not the way it's done. But I never intended to practice at all. Never the slightest intention to practice.
I'm sure you'd have great bedside manner.
Well, I'll tell you, the chair of psychiatry came to me when I was graduating and said, why don't you go into psychiatry? You'd be absolutely fantastic.
Oh my God, you'd be awful.
Anyway, it's all in the eye of the beholder, right? Yeah.
Lee, to stay on the topic of medicine for a little bit, you've been thinking about medicine and the applications of biology to medicine for quite a while now, and I guess what's your current view of how this is going to play out? And you made an interesting parenthetical comment earlier in the evening that deliverables convince skeptics. So what are going to be the P4 deliverables that are going to convince people this is a viable way forward and a useful way to pursue medicine, both from the standpoint of biology and therapeutics or disease prevention?
Okay, so I'll answer that two ways. I'll give you a list of the things I think P4 medicine will really bring to medicine, very, very concisely. And then I'll tell you how we're going about doing that. So I think this systems approach to medicine, one, is going to give us really detailed mechanistic insights into disease that we never had before. And we've done 2 mouse model systems with the dynamics and all the rest of the things, and I can tell you in detail about how that's worked out really well. I think number two, something I'm really excited about is the fact that, so I think in 5 years, your genome is going to cost $200 or $100. And I think in 10 years, genome will be a routine part of medical record. And the reason I think that is the driver will be actionable variants. That is, variants when identified for the individual, physician can tell them something that'll change their life. For example, with something like Lynch syndrome, where the mutation is a dominant, and it's in one of the enzymes that deals with DNA repair. And what do you do about that? You have them start colonoscopies at 25, and you just snip out the crypts at the time, and you can actually keep people alive that way. So I think, and one of the things that's going to be great about the genome is once you've done that, then the number of actionable SNPs variants are going to increase exponentially, so your genome will be searched every year. And in a sense, your genome is an investment in optimizing your health for the rest of your life, if you want to look at it that way. So that's the second thing I think. Now, can systems biology claim credit for the genome and actionable variants? I'll leave that up to you. I think a third thing that systems biology has already started to do is it's going to make blood a window into health and disease. And it can do this in many ways, but one of the coolest ways is we've demonstrated that most organs have organ-specific proteins, a fair number of which are secreted into the blood. So there they constitute a brain-specific fingerprint. And if you have a normal brain, the levels of those proteins will be set at one series of levels. If you have a diseased brain, then the proteins whose cognate networks have become disease perturbed will change their levels. So you'll get, for each different disease, because they perturb different combinations of networks, you'll get a unique fingerprint. And we've really demonstrated all of these things very beautifully. And the other thing that organ-specific fingerprints are going to do well is we're really pushing with Novartis the idea of zero-phase drug toxicity tests. That is, you take something whose safety and efficacy have been established, and you give them at very low levels to patients, and then you just interrogate the major organs of toxicity, liver, heart, kidney, wherever. You can decide what you want to look at. I think a fourth thing that systems biology is going to do is it's going to give us really effective tools for the stratification of disease. So, and that's really important because that stratification is going to be utterly essential to a proper impedance match against the right kind of drugs. And even more, one of the things we're really excited about is we're going to be using iPS cells in clever ways to stratify Alzheimer's. And isn't it going to be great to go to the drug companies collectively with their, 130 drugs for Alzheimer's, none of which have been worth a hill of beans, and say, look, we have 8 stratified populations. Test your drugs against those, and if they're going to work, they'll work in something that's been stratified. I think a fifth thing is that you can also use those organ-specific fingerprints to look at multiple organ responses to disease. And that's essentially a network interaction level that's way above genetic, molecular, cellular. It's up at, and I think that's going to be very important in disease for the future. I mean, diseases aren't single organ diseases, any of them virtually, and you'll want to know what the other organs, how they're responding. I think a sixth thing is that we're going to provide a really interesting new way for choosing drug targets. So you know that what drug companies are really great at is making drugs. What they're terrible at is choosing the choice of targets for drugs. And so the idea in systems medicine is that we will be able to use drugs to re-engineer networks that have become disease perturbed to make them behave in a more normal fashion. And we've shown in one of our studies very beautifully that, for example, the 4 major networks in a certain neurodegenerative disease in mice are sequentially disease perturbed. So you go for the most proximal one and see if you can abrogate that disease perturbation, then you'll shut down all the downstream kind of things. And so what we're doing now is going back into microorganisms to learn the rules of re-engineering networks with drugs. I mean, and it's not an easy thing. It will certainly take a long time. I think another thing that's really going to be important is that, well, one of the big revolution that's coming in medicine is the quantization of the self. We're going to have enormous numbers of parameters digitalized and measured in the future. And they'll be the common kinds of things that you know and think about. There'll be enormous molecular things, and they'll go all the way up to what comes to you from your social network interactions and things like that. And I think that digitalization of information on the self at the level of all these interacting networks, again, genetic networks, molecular networks, cellular networks, organ networks, individual networks, social networks, that's going to give us deep insights into optimizing wellness for the individual. And in fact, one of the things we're thinking about now that is, and I think there's a real shot we have at doing it, is we'd like to get blood metrics for wellness, not only that indicate in some relative sense whether you're well or whether you're sick, but they give you an indication of the slope of wellness. Are you going in the right direction or away from the right direction? So those are some of the things that I think are really going to be transformational with P4 medicine. And so how are we going to go about bringing it to patients? So what we've done is created the P4 Medicine Institute, which is a nonprofit institute whose mission is to collaborate with ISB in creating a network of clinical centers. And the objective with these clinical centers is for each of them to take on a couple of pilot projects that will use these strategies, thinking, tools of P4 medicine. So the first 2 partners that we've recruited are Ohio State Medical School. In fact, I just came back this morning from there, and we've decided on 2 pilot projects, and I'm really excited about both of them. One is wellness, and we can talk about what that means, and the other is preterm birth deliveries. It's one of the biggest costs to healthcare in the world, and I think there are really effective ways for beginning to figure that out. So that's what we want to do with them. And then Peace Health is a community hospital in the Northwest that has 5 major hospitals, and we're probably going to take a very large series of families and begin measuring a whole series of parameters that, again, deal with aspects of wellness in those families. And the idea is that what I'd really like to see, and I will say we've got at least 4 other medical schools that are really interested in this, and Cleveland Clinic and MD Anderson, and there's a wonderful community hospital called Inova in Northern Virginia that is really doing some pioneering things, which I can tell you about. But what I'd like to see is in 2 to 4 years that we really have a couple of pilot projects that are spectacularly successful. And then the question is, where would you go next? And my vision is we go to a small country. See, I think the U.S. is utterly impossible to really try and introduce to new ideas. I think what you have to do is show it to the U.S. from the outside. And I've been accused of being anti-patriotic for this, but I mean, we're heterogeneous in our healthcare systems, on and on and on. But a place that we have an agreement with Luxembourg, and that's one thing that we're beginning to explore with Luxembourg. And, you know, there are other places that are really could be interesting. I was just at Qatar, and I think that might be a very interesting place to look in the future. So they have all the money to do it, and I think in many ways they have the will to do it. What they don't have are the scientists to do it, and I'm not sure how you get around that in the short term.
We better end, I think. Thank you all for your attention.
Okay.